All right, so without further delay, let's start with today's webinar. So hello everyone, a very warm greeting to one and all. My name is Srigauri Krishnakumar, and I would like to welcome you all for the first session on bioinformatics for infectious disease program. This program is dedicated to the study of viral diversity and its role in epidemic infectious diseases that keep re-emerging, including zoonotic spillover, transmission between humans, and the process of viral and bacterial disease development. And in the first session, we will gain an overview of next generation sequencing, wherein we will cover topics on read sequences, file formats, alignment, annotation, and non-map reads, and alignment to databases of viral genomes. So before we begin with today's session, for those of you who are joining with us for the very first time, let me take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. And as a part of our mission, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. And this training has been completed by over 24,000 participants from 187 countries in over 300 workshops in 6 different specialization tracks that includes oncology, infectious diseases, precision medicine, neuroscience, and data science for biomedical data. And as a part of the training, you will have access to the OmicsLogic resources, which takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path with the Bloom's taxonomy pyramid. And this also comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics that includes genomics, transcriptomics, metagenomics, and epigenomics, data science that makes use of bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. And the OmicsLogic platform also gives options for analyzing a given dataset. These datasets and associated publications can be used in several ways. That is, you can learn bioinformatics without any coding at all. For those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run the bioinformatic pipeline. And different stages of analysis are performed in different sections on this multi-omics platform. And for those interested in learning to analyze data through coding, we provide the OmicsLogic Code Playground, which will help you develop coding skills while learning popular packages in R programming and Python to visualize, annotate, and analyze complex patterns in biological data. So now, to view the various courses, example projects, you need to first sign up on the OmicsLogic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features that you need to update on your account. That is, a completed profile will have a profile image, a full name with appropriate capitalization, link to your social media handle, a brief bio about yourself that will consist of your educational background and research interests. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. Finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the OmicsLogic Learn portal. So those of you who have not signed up yet, kindly sign up. I'm pasting the link for signing up in the chat box. So please visit the link and I'll show you how you can sign up on the portal. So for those of you who have not signed up, please click on the Create an Account option that you see here. Once you click on this, you'll be able to enter your name, email ID, and password, and you can sign up. Or you can also sign up using any of your social media handles. This could be Google, Facebook account, Apple account, GitHub account, or even Twitter. And for those of you who have already signed up, you can simply click on the login now option and enter your existing email ID and password and sign. So with that, I'll pause for a couple of seconds for the participants to sign up and sign in. And once you have, please put a yes in the chat box so that I can proceed ahead. We are getting few responses from Robert, Dr. Sudhir, and Nurena. Thank you so much. What about the rest of you? Have you also signed up? Thank you so much, Uluvaimi. Thank you, Harveet. Thank you, Karthik. All right, so since the participants are signing up, I'm just going to proceed ahead. Along the way, if you're facing any technical challenges, please let us know in the chat and we'll be happy to guide you. So since I already have an account on the portal, I'm going to directly click on sign in. So once you sign in, you will be visiting the courses page. So here you'll be able to see the various courses and example projects that are there on the portal. And once you scroll below, you'll be able to see a search bar that says looking for a specific course. So here, if you enter a keyword or research interest, then that will show you the various courses and example projects that makes use of that call. For example, since a lot of the participants are interested in infectious diseases, if you enter the keyword infectious disease, it will show you the various courses and example projects that range from tuberculosis infection, malaria infection, SARS-CoV-2, and so on. So the participants can take a look at all of these at their own pace. I just wanted to show you how you can navigate the courses tab. And coming to the part where you have to set up your profile. For that, you can click on this blue bar that is welcome back with your email ID. When you click on this, it will directly take you to your account setting. Here, when you will see an icon that says profile, that will enable you to edit all the features that I was talking about in my presentation. That is, you should have a full name, appropriate capitalization. You should add a bio that consists of your education background and research interest, add a profile image for yourself, as well as link your social media handle. So that was about setting up the profile. And then you'll also see several tabs under your profile. Under the activity tab, like I mentioned, the program mentor and the community manager, they will be tracking your learning progress. The courses that you're currently taking will show up under the courses tab. Once you complete a research project on the program on the portal, that will be displayed under the projects tab. And once you enroll for the program progress can be viewed under the program tab. Finally, after the completion of your coursework with the required quizzes and assignments, the certificates will appear under the certificates. So another important tab that I want to show you is the projects tab, which will show the various student projects that have been completed as a part of several of the programs. And the topics range from infectious disease, precision medicine, neuroscience, machine learning, data science, and so on. So the participants can have a look at all of these student projects as well at their own pace. So that was all about navigating through the portal. And with that, now I'd like to pass on the stage to Sonalika, who will be starting with the session. So over to you, Sonalika.
Thank you so much, Shrigari. Can you please confirm if my screen is visible?
Yes, we can see your slides.
All right, thank you so much. Good evening, everyone, or good morning and good afternoon in whichever part of the world you are. Today, we will discuss and proceed ahead with the first session of the program, Bioinformatics for Infectious Diseases. We have designed this program to introduce the data, resources, the tools that you can use to apply bioinformatics to publicly available data on infectious diseases. In this program, you will understand the viral diversity and its role in epidemic infectious diseases that keep re-emerging, including zoonotic transmission, transmission between humans, and the process of viral and bacterial disease development. We learn about bioinformatics analysis approaches to study viral genomes and understand the relationships between viral strains, haplotypes, finding differences in sequence data, and seeing the implications for drug and vaccine design. You'll gain the hands on experience by analyzing curated datasets from publicly available domain collections guided by experts with bioinformatics experience and knowledge about virology. We'll also address some questions about infectious diseases, like what is a pathogen, where do diseases come from, what causes their diseases, how do these diseases spread, how to treat and or prevent the diseases, and what remains to be done. Global survey shows that in 2019, antimicrobial resistance killed more people than HIV or AIDS or malaria deaths. From drug-resistant tuberculosis account for about one-third of all antimicrobial resistance deaths worldwide. Before we get started, let us discuss what an infection is and what are these different types of infectious diseases. So an infection is the invasion of an organism's body tissues by disease-causing agents, their multiplication, and the reaction of host tissues to the infectious agents and the toxins they produce. An infectious disease, also known as transmissible disease or communicable disease, is an illness resulting from an infection. Infections are caused by infectious agents called pathogens, which includes viruses and related agents such as viroids, bacteria, fungi, parasites. Unicellular organisms, that is malaria, toxoplasma, babesia, or macroparasites like worms or helminths, including nematodes such as parasitic roundworms and pinworms, tapeworms, and flukes as well. Non-infectious diseases are also recognized as chronic and can be caused by such factors as environment, nutrition, and lifestyle, as well as genetic inheritance. WHO recognizes only four main types of chronic and non-infectious diseases, which are cancer, cardiovascular, chronic respiratory diseases like asthma or chronic obstructive pulmonary disease, and diabetes. Infectious diseases are caused by pathogenic microorganisms such as bacteria, viruses, and parasites. There are many different types of infectious diseases, some of which are common than others, like respiratory infections, including common cold, influenza, pneumonia, tuberculosis, gastrointestinal infections, including gastroenteritis, cholera, and hepatitis A, sexually transmitted infections that are spread through sexual contact, including chlamydia, gonorrhea, syphilis, HIV, and AIDS, vector-borne infections that are transmitted by vectors, such as mosquitoes, ticks, and fleas, zoonotic infections that are transmitted from animals to humans, such as rabies, Ebola, and bird flu, or skin infections, which includes impetigo, cellulitis, and ringworm, or blood-borne infections that are spread through blood, such as hepatitis B and C, and HIV or AIDS. And then we have fungal infections and parasitic infections as well that are caused by fungi and parasites, respectively, which could include ringworm, candidiasis, malaria, trichomoniasis, or toxoplasmosis. It's important to note that some infectious diseases can fall into multiple categories, and some disease can be caused by more than one type of microorganism as well. The replication of pathogens in the host leads to major interference, starting from membrane disruption, as in the case with viruses, and the release of toxins, as in the case of many bacterial pathogens. Many of these interferences induce molecular biological pathways designed to fend off intruders and signal to neighboring cells of danger. The pathogens can also modulate these pathways and cause long-term organ damage, such as lung tissue, scarring due to the immune hyperactivity. Now, to treat these pathogens, vaccines were designed first, as far back as the early 1800s, and were becoming major instruments to train the human immune system in response to various pathogens, reducing worldwide deaths significantly in the 20th century. As a result of water treatment, use of vaccines and antibiotics, death rate from infectious diseases was reduced tenfold in the 20th century, but in the 21st century came around with the new challenges, antibiotic resistant bacteria, and new challenge from novel pandemic viruses that we were not prepared for. Thankfully, at the same time that the pathogens were studied under the microscopes, humans also started developing sequencing technologies that could help decipher the human genome. As a result, such detailed and rich molecular data has transformed our understanding of infectious diseases. In the last 20 years, availability of genomic data has led to an increasing understanding of how cells function at the molecular level and has allowed us to understand major causes of deadly diseases. With the current rise in viral outbreaks and their deadlines, there is a major focus on developing tools and data repositories for a better understanding of the emergence, prevention, and treatment of infectious diseases. As a result, the major advances in data generation, analysis methods, and utilization of insights from such studies is making a completely new approach to diagnostics, disease characterization, and development of interventions. Genomics and bioinformatics have contributed immensely to our understanding of infectious diseases, from disease pathogenesis, mechanisms of interaction, the spread of antimicrobial resistance, to host immune responses. As a result, the emergence of genomic medicine has substantially advanced our ability to detect, study, and treat infectious diseases. These achievements came as a result of a close collaboration between computer scientists and biologists, placing bioinformatics at the center of current advances in this domain. As a result, omics data is now regularly used to study viral genomes, DNA variation, host gene expression, protein abundance, and composition properties of microbial communities, all very different types of data that we need to effectively study and mine for patterns to understand host pathogen interaction. The role of bioinformatics in general and in the context of infectious diseases is to allow researchers to access, process, analyze, and visualize these kinds of high throughput data. And today I will introduce several examples of how this was done successfully by researchers studying viral infections like the Ebola virus outbreak in West Africa, COVID-19 pandemic, as well as microbial pathogens causing tuberculosis and single cell parasites that cause malaria. We will briefly touch on viral genomics and evolution, gene expression patterns, and detection of significant genomic variants, as well as the important aspects of microbiome and virome communities in a host that are being studied to find important association with disease susceptibility and outcomes. The virome is one part of the microbiome which constitutes all of the organisms that inhabit us, including bacterial and archaeal organisms, as well as the mycobiome and other members of myofauna, such as protists and metazoans. Studies of the virome are in their infancy because it has only recently become possible to see the virome in large sequence datasets using bioinformatics tools that can detect relationships between viruses despite extreme nucleotide sequence diversity. The size of the mammalian virome is not known, although our own cells are outnumbered about tenfold by our bacterial microbiome. The number of viruses may be tenfold higher. Still, human feces alone contains 108 to 109 viruses per gram. Additionally, the mammalian virome is continuously updated through rapid evolution of viruses and through exposure to viruses in the environment or in other mammals. Now, here is the scale of sequencing information that is available in GenBank. The first graph here shows the percentage growth of sequences deposited on GenBank between August 2009 and August 2010. Sequences are grouped based on the GenBank classification of division, either by their high taxonomy or the sequencing method employed. And the graph on the right side shows the 20 most frequently sequenced viruses appearing on GenBank. Sequences were identified here based on their organism name as described in each submission. Thankfully, this specific outbreak brought about a huge collection of data. This information is becoming widely accessible. Millions of sequences and hundreds of thousands of full genomes are available on repositories hosted by the National Center of Biotechnology Information and other databases like the Joint Genome Institution's Integrated Microbial Genomes Database. These databases are growing in size at an exponential speed. Many different sequencing techniques were introduced over the years. Sequencing platforms use a variety of read lengths. The benefit of short reads isn't that they're short, but that they can be very inexpensive and can be generated in a massively parallel manner with billions being generated in a single run, where cost needs to be kept down and the read counts must be high. The short read technology makes the most sense. Long reads, on the other hand, can resolve regions of the genome that are inaccessible to short reads due to repeat sequences. NGS allows researchers to explore genetic variations like never before, down to a single nucleotide resolutions, whether you need to identify specific variants from a focused set of regions or look more broadly for potential causative variations. Whole exome sequencing or whole genome sequencing both offer effective solutions. Whole exome sequencing is a targeted view of the protein coding regions of the genome. It offers reliable and sensitive detection of coding variants. It's fast and cost effective. Whole genome sequencing, on the other hand, provides a comprehensive view of the genome as far as the coding, non-coding, and mitochondrial DNA. It's reliable and sensitive as well, but includes the detection of all the variant types. It's low cost and a fast method of library preparation as well. We will learn about the sequencing process. Transcriptome sequencing is a major advance in the study of gene expression because it allows a snapshot of the whole transcriptome rather than a predetermined subset of genes. Whole transcriptome sequencing provides a comprehensive view of a cellular transcriptional profile at a given biological moment and greatly enhances the power of RNA discovery methods. And to study this data, we will learn about processing the pipelines in our next sessions that turn raw reads into structured data that is ready for analysis. We'll also discuss about metagenomic and viral sequencing techniques and processes in the next sessions. We'll learn how a genome is assembled and referred for analyzing data from different resources to biological insights for designing a better therapeutic approach. When dealing with infectious diseases, different types of data may be collected and analyzed to better understand the disease and its transmission patterns, such as epidemiological data, which includes information about the incidence, prevalence, and distribution of the disease in a particular population, which includes age, gender, and geographic location. Or it could be clinical data. This includes information about the signs and symptoms of the disease, as well as many medical tests or treatments that are used to diagnose or manage the disease. Laboratory data, which includes information about any laboratory tests or analysis that are performed on samples from patients, such as blood or tissue samples, to detect the presence of the pathogen causing the disease. Or it could be environmental data, which includes information about the physical environment in which the disease is occurring, including factors such as temperature, humidity, and pollution levels. Genetic data also includes information about the genetic makeup of the pathogen causing the disease, as well as the genetic makeup of the host population, which can be helpful to identify any genetic risk factor that may be associated with the disease. Apart from this, you also have social and behavioral or economic data. And by analyzing these different types of data, researchers and public health officials can gain a better understanding of how the disease is spreading, what factors may be contributing to its spread, and how best to control and prevent the transmission. In the next sessions, we'll deal with each one of them. Let's now discuss about different NGS types, advances, and the applications of Illumina sequencing technique, nanopore, and PacBio sequencing technique. Some of you might be from an undergraduation or a graduation level and a different background as of bioinformatics. So we quickly wanted to go through these different types of sequencing techniques and methods to give you a good background. So let's start with the first sequencing technique, Illumina, that is widely used DNA sequencing technology that allows for high throughput and low cost sequencing of data. It uses a process that is called sequencing by synthesis, where nucleotides are added to a growing strand of DNA and their incorporation is detected by fluorescent dyes. The resulting data is then used to generate a sequence of DNA-based pairs. Illumina sequencing is commonly used in many areas of biology, including genomic research and personalized medicine. Nanopore sequencing is a technology for DNA sequencing that works by passing a single strand of DNA through a protein pore, with each nucleotide causing a change in the ionic current as it passes through. And these changes are detected and used to generate a sequence of the DNA molecule. Whereas the PacBio sequencing is a single molecule real-time DNA sequencing technology that uses polymerase enzymes to synthesize long strands of DNA, which are then imaged as they pass through a zero-mode waveguide. The sequencing process generates long reads with average read lengths, typically ranging from 5 to 15 kilobases. So what are the different types of genomic sequencing techniques? Let's understand the basics of targeted, whole genome, and whole exome sequencing. The targeted sequencing is a method of DNA sequencing that focuses on specific regions of interest in a genome. It is more efficient and cost-effective alternative to whole genome sequencing and can provide higher coverage and accuracy for specific genes or genomic regions. Whole genome sequencing is the process of determining the complete DNA sequence of an organism's genome. It provides a comprehensive and high-resolution view of the entire genome, including both the coding and the non-coding regions. Whereas whole exome sequencing is a type of DNA sequencing that focuses on the exome, which is the portion of the genome that contains exons. Exons are the parts of a gene that contains the instructions for making a protein. Now let's discuss about different technologies for DNA and RNA sequencing. Again, just going through it to briefly have an overview for beginners, which includes microarray sequencing, high-throughput sequencing, and qPCR. Microarray sequencing, which is also known as DNA microarray or DNA chip technology, is a method of analyzing variations in DNA sequences. High-throughput sequencing, also known as next-generation sequencing, is a powerful technology for large-scale DNA sequencing. It enables the rapid and simultaneous sequencing of millions or billions of short DNA fragments, providing a high-resolution view of the genome. This technology has revolutionized the field of genomics and has a wide range of applications, including genome sequencing, transcriptome analysis, epigenetic studies, and metagenomics. The quantitative PCR technique, the last one, is widely used molecular biology technique for measuring the amount of DNA in a sample. The qPCR is a real-time quantitative method that enables the measurement of specific DNA sequences in a sample by monitoring the amplification of DNA in real time as the reaction progresses. These different steps of genomic data analysis typically involves several file formats, including, let's start with the first one, the raw data, which typically is in the FASTQ format for sequencing data. The next step, that is quality control and pre-processing, where we have trimmed and filtered reads that are stored in FASTQ format. Quality control reports are stored in PDF or HTML format. The next step for alignment, the reads are aligned to a reference genome in BAM, that is binary alignment map format, which is a binary version of the SAM format, SAM, sequence alignment map. The next step for variant calling, where the variants are stored in VCF, variant calling file format, which contains information about the location, type, and quality of genetic variations. And then comes the annotation, where variants are annotated with functional information stored in GFF or GTF format. GFF stands for general feature format, GTF stands for gene transfer format. Now, different analysis of the pipelines may require additional file formats such as BED, that is browser extensible data for interval analysis, or WIG, Wiggle format for continuous data like chip-seq signal tracks. It's worth noting that many bioinformatics tools and pipelines will automatically convert between these formats as needed for different steps of analysis. In the next sessions, where we will have a practical hands-on with different types of data, we'll run these pipelines with different types of data files and have a look what these files include and how to interpret the results that we get from the different files like a variant calling file, or how do we annotate them. The sequencing data is stored in several different file formats that I just mentioned. Let's quickly have a look at each one of these file formats. So let's begin with FASTQ. FASTQ provides us with the reads from the sequencer. It is a text-based file format that stores nucleotide sequences of reads and quality scores for each letter. FASTA is a text-based format for representing DNA and protein sequences. Typically, this is the reference file for the genome. Then we have the GTF file, which contains genome annotation. For example, the GTF will store the start and the end of each genomic element, exon, isoform, and gene. Genomes are assembled from next-generation sequencing techniques, which is typically done using a process called shotgun sequencing. The process includes shattering the source material, RNA or DNA, and then sequencing short reads of 30 to 300 base pair long. These sequences are assembled into a full genome by looking at the consensus sequence. In other words, if the sequence of the reads all shows the exact same letters, they are considered the consensus. If there is a disagreement, the majority wins. As a result, from a single isolate, we can get the full sequence of a viral genome that is most abundant. And this process is fairly accurate. However, it reduces the diversity of potential present variations of the virus in each sample to a single sequence. The major sample sequenced, the higher is our confidence that a strain has consistent characteristics. And after these sequences are found, we can start looking at the genome characteristics such as genes or special coding sequences that can be associated with a function. Let's first speak about the sequence itself, like FASTA. FASTA is commonly used file format for representing nucleotide or protein sequence data. It is a plain text format that includes a header line beginning with a greater than character, followed by one or more lines of sequence data. FASTQ is another commonly used file format in bioinformatics, particularly responsible or particularly represents next-generation sequencing data. It includes both the nucleotide sequence data and quality scores for each base. Then we have the BAM and the SAM files that are file formats used in bioinformatics for storing and exchanging aligned sequence data, particularly for data from next-generation sequencing experiments. SAM is a text-based format that stores aligned reads in a tab-delimited format. Each line of a SAM file represents an individual read and contains information such as the read ID, its alignment position, its mapping quality, and the actual sequence and quality scores. The SAM files are human readable and can be edited using a text editor. However, they can be quite large, particularly for NGS data, which can result in slow processing and high storage requirements. Whereas the BAM files, they are compressed binary version of the SAM format. It stores the same information as the SAM file, but in a binary format that is much more compact and can be processed much more quickly by bioinformatics software. The BAM files are generated by compressing the SAM files using the SAM tool software, and they are preferred format for storing the NGS data due to their efficient storage and processing. Both the BAM and the SAM files, they include information about the aligned reads, including their mapping position, strand quality score, and any potential mismatches or indels compared to a reference sequence. They are used for a wide range of downstream analysis applications, which includes variant calling, gene expression quantification, and functional annotation. In this program, we learn to visualize the sequence as they are mapped on the reference genome and look at the variants of interest while annotating them using databases like Glenvar, DBSNP, and Cosmic. As a result, we will see how important nucleotide level variation can be studied from the codon level to study the variant types for single and multi-nucleotide variation. We'll also focus on processing and analysis methods that are used to analyze and integrate genomic and transcriptomic data. We learn about the analysis methods and tools for genomic and transcriptomic data analysis of host pathogen interaction, the methods to study relationship between or the relationship based on local and global multiple sequence alignment, phylogenetic analysis, reconstruction of a tree, and evolutionary studies that are used for genome ancestry and population fitness. We'll also discuss about chemical and structural implications of sequence variation mapped on the PDB models and discuss the immunology of host pathogen interaction and pathway analysis of treatment response, where we will consider common considerations for accurate discrimination between statistically significant and insignificant variants at the read level, as well as advanced methods to deconvolute, such as coverage variation in large data sets. We will also discuss methods for differential variant calling that can help us compare multiple samples for associations with phenotype. Ultimately, we will find the way such studies can help explain captured data from the point of view of a mechanistic model of replication, immune response, and disease pathogenesis that all contribute to the variability between and within each sample based on but and single cell data. We'll also construct phylogenetic trees, tracing how the observed species we capture can represent a common source origin and even evolutionary time that separates multiple samples in a study. Such phylogenetic studies have yielded important information on how a pandemic or even disease of a single individual cages with time and gives rise to novel pathogens. We will use the example of viral outbreaks like the Ebola virus epidemic of 2014-2016 and the ongoing COVID-19 pandemic. Relying on easy-to-use tools for multiple sequence alignment and annotation databases that show the organization of elements in the viral genomes, we will be able to replicate the key findings referenced in top peer-reviewed journals on the emergence and evolution of SARS-CoV-2. We will learn about different public repositories that you can leverage, extract the data from, and for further analysis. We will look at the variability of nucleotide sequences between all the coronaviruses to find specific genes or even the regions that are less conserved and discuss their functional significance. We'll then go beyond sequence analysis, where 3D structures and transcriptomic data can help us understand the implications of genomic variants that we identify. As we see here, similarity scores are especially low in the spike glycoprotein, which is responsible for the attachment of the viruses to the host cell. We'll also further do an evolutionary analysis of genome sequences from the 2013-16 epidemic that have provided a clear picture of the origin and spread of Ebola virus. One of the most important early questions was whether the epidemic was the result of a single cross species transmission event into humans or whether there were repeated zoonotic events from a widespread animal above reservoir. Due to the high genetic similarity of virus genomes sampled from the beginning of the epidemic, a single spillover infection seems to be most likely the case. Phylogenetic analysis also makes it clear that once the outbreak was established, later lineages of above Macona have descended from those circulating earlier in the epidemic, and this is in contrast to some of the earlier EVD outbreaks in which the epidemiological and sequence-based investigations have provided evidence for multiple spillover infections. Here we review the methods for the analysis of HDS reads, including approaches to local diversity estimation and global haplotype reconstruction. Challenges posed by aligning reads as well as the impact of reference biases on diversity estimates that are also discussed. In addition, we address some of the experimental approaches designed to improve the biological signal-to-noise ratio. And in future, the computational methods for the analysis of heterogeneous virus populations are likely to continue being complemented by technological developments. Taking this into regards, we'll also do a hands-on with these different softwares where we can visualize these hosts and understand the host pathogen interaction. The heterogeneous mixture of viral strains appear to confer numerous advantages to the virus population, which includes the ability to escape from the host immune response and the development of resistance to vaccines and antiviral drugs. Furthermore, the existence of different viral strains have significant implications for viral pathogenesis, virulence, persistence, and disease progression, and likely contributes to tissue tropism. The robust adaptability that is featured by RNA viruses, which is related to the genetic heterogeneity, is thus of clinical relevance that we would discuss. In fact, many of the infectious diseases which have jeopardized and still are a threat to public health are caused by RNA viruses, including HIV, HCV, and influenza virus, Ebola virus, and also Zika virus. We explore some case studies as well as we proceed in the program, such as malaria, SARS-CoV-2, Ebola, EVD68, and tuberculosis. Tuberculosis is a contagious infection that usually attacks the lungs. It can also spread to other parts of the body like the brain and the spine. And a type of bacteria called Mycobacterium tuberculosis causes it. In the 20th century, TB was a leading cause of death in the United States. And today, most cases are cured with antibiotics. Also, we'll go through some publications and case studies throughout the training program. And these methods will include hands-on exercises on the T-Bioinfo platform, which provides a simple and intuitive user interface for processing, analysis, and machine learning methods for multi-omics data. And towards the end, you can also work on your project under mentor guidance by applying the GAIN skill set from the training program. So that is all that I wanted to discuss today. I just wanted to get you all started with discussing about some of the NGS techniques and also what we'll go through in the next sessions of the program. I would now like to take you through the OmicsLogic Learn portal and the program page to discuss about what are we going to cover in the next sessions. We didn't want to overwhelm you a lot, so to get started, I think we have a good beginning. If you have any questions, you can please put in the chat box. I will now share my screen to have my Shri Gauri, can you confirm if you can see the OmicsLogic Learn portal on the screen?
Yes, we can see the portal.
Thank you, Shri Gauri. All right, so now we are here on the OmicsLogic Learn portal. I would refer to the program page and I will also tell you which all coursework or resources are associated with this program. So once you click on the programs tab, if you lose a track of the program page, you can go to the portal, click on the programs tab, and here you have this program which says Infectious Diseases for Bioinformatics Research. You need to click on this, and when you click on the program, you will get the program page. Just a second. Yeah, all right. Now, once you are here on the program page, the registered participants can see on find the program page badge on their profile. I don't have the badge right now on my profile, but those who have enrolled for the program, they should see the badge of infectious diseases on their profile. If it's not there, please tell us in the chat box so that we can add one. It's important to have this badge so that you can access the recordings after the live session and also go through the associated resources that are mentioned here. Okay. And now once you register for the program, for the registration, how do you register for the program? I'll pass on the stage to Shubham. For the first session, we have invited all the interested participants who wanted to join the infectious diseases program so that you get an idea of how the program would be conducted online via Zoom classes and what the structure of the program would be like. Now, once you're on the program page and you scroll down, you can find the schedule that includes all the details of the sessions that would be held, on which all dates the sessions would be held, and it would be usually around the same time as today's 8:30 p.m. IST or 9 a.m. CST. And here you have the topics for each of these sessions. Now, with the topics, you can see some associated resources. These are actually courses or lessons of the courses on the OmicsLogic Learn portal. They all are hyperlinked. If you click on it, you will get redirected to a specific lesson of the course that you need to go through. All right. And here you have a mix of context, images, videos to supplement your knowledge. And towards the end, you have a quiz that you have to answer. I'll also take you to the source of these courses so that you have the access and know where the resources are present, and you can go through it otherwise separately too. The next session would be held on Feb 21st, where we'll discuss about multiple sequence alignment and phylogeny. The structure of the program is such that we will first of all discuss the concepts first, and then we'll take you on the T-Bioinfo server, and then we'll practice the pipelines. We'll do a hands-on, and we also have some R lessons for multiple sequence alignment, phylogenetic analysis, etc. We'll showcase the resources to you as well. The R lessons are though not included in the program, but if you're interested to learn the computing language R for carrying out all these different types of analysis with infectious diseases, you can go through those lessons. All right, so you have the topics mentioned here. You have the program. I'll just put the program page link in the chat for you. Yes. So you can go through each one of these topics. I'm not actually reading all the topics right now. We're here just telling you about the sessions. The next session would be about multiple sequence alignment and phylogeny, and then we'll have a hands-on session where we'll take up the case study of COVID, and we'll also discuss about Ebola briefly. We'll have session where we'll just discuss about the pipelines that you all process and then look at your results and clarify your questions. And then we'll discuss about how infection leads to pandemics and how the transmission happens. We'll discuss about symptoms, severity, disease progression, and the outcomes. And we'll also discuss about different case studies, rate of mutation, mutation annotation, and that is significant for analysis, and host pathogen interaction. So we'll be going through all of these different topics throughout the program, and just showing you the T-Bioinfo platform to answer some of the questions and curiosities by the participants here. So we have two platforms. One is the OmicsLogic Learn portal, that is the training platform. You will learn from these different courses on the OmicsLogic Learn portal. Specifically for this program, we'll be taking you through the genomics course and the metagenomics course as well. And these are the courses that you will go to build up some concepts and also to learn how different analysis could be carried out using the T-Bioinfo platform and also through computing languages like R. And then we'll also go through some case studies to understand how some research questions could be answered with the skill set that you gain throughout this program. Now, when I say these research studies, you can click on the project tab and then infectious diseases. We have case studies on COVID, Ebola, EVD68, Spodoptera, frugiproda, and then a surveillance of SARS-CoV-2 as well. We'll go through some of these case studies and try to understand the emergence of these different infectious diseases. We'll try to compare the different strains, and also we'll try to understand the pathogenesis and some transcriptional signatures, some biomarkers that we could identify for the treatment. Now, coming back to the T-Bioinfo platform, I wanted to mention those who have enrolled for the program. You might have received an email by Sri Gauri. For the OmicsLogic Learn portal, your credentials would remain the same that you would create for yourself. But as a part of this program, you get an educational premium license on the T-Bioinfo server. And to activate the same, we change your passwords for the server. So you will get the credentials for the server. Your login ID will remain the same as your email ID with which you signed up, and your password would change. And using the set of credentials that we send you via your email, you should be able to get an access to these pipelines. Now, these are the two sections that we'll go through, the genomics one and phylogenetic analysis one. And if you click on these drop-down buttons, you have some demo pipelines that you can run through and see what types of results you get and how do you interpret them. Otherwise, in the program, we will explore data from different data repositories. We'll extract them, we'll prepare the data, and then we'll run these pipelines. And when we run these pipelines, we'll use some different tools or algorithms for data pre-processing, mapping, visualization, meaning calling, and annotation, what these algorithms are, what do they do, how do they process the data, what output you get, how do you interpret the output. There are a lot of things in this workflow to answer. So we'll look at each one of these different steps one by one throughout these sessions. And also to supplement your knowledge, we have the content in the coursework that you have to go through as a part of each and every session. And also you have to practice these pipelines and then interpret the results. And then here is the phylogenetic analysis pipeline. As today was the first session, I just wanted to briefly give you an overview of what is inside the program, what we learn, what pipelines will be run, and also what is the access time, so that there are no more confusions. And also after the live sessions, once the recording is processed, we'll upload it on the program page. So you can revise the concepts, go through the recording once again on the program page. I'll show you some other program where we have already uploaded the recording so that you can see how the recording comes up. If there are any questions, you can put them in the chat so that we can address. That was it that I wanted to mention. If you have not registered yourself for the program yet, you won't have the badge. Once you register, then you'll have the badge. Olva Yemi, I'm not sure if we have received your registration. Our team will check and then would get back to you on this. I'm just opening up a program page that is ongoing to show you how the recordings are uploaded. So this is on the program page, you have the recording like this, and then you can go through it. Now that was it. I would stop sharing and would pass on the stage to Shubham to help those who are interested to enroll for the program but have not registered now.
All right, thank you so
Shubham,
much.
over to you.
Thank you so much, Sonalika. That was a very wonderful and informative overview session, we can say. Hello everyone. I'll just share my screen. I'm Shubham. I'll be like briefing you about the enrollment and the registration part for all those who are like interested to like go with this program. Uh, Snalika, is could you
Shubham,
confirm?
just a second. I can see a question by Rajeshwari. We have 11 sessions that you have to complete as a part of the program to complete it. Shubham, over to you.
All right. Is my screen visible?
Yes.
Okay. Like as you all can see and like mentioned by Sonalika, that this is a program page for the infectious disease bioinformatics research and applied genomics, and it will like the like course has already been commenced from today, like Feb 17 to 30th April. So furthermore, like if you scroll down, you will see that there is a program overview section. So what all topics you will be getting to learn and what all concepts you will be going while this program, and like some of the hands-on sessions will also be covering. So all these parts we have covered in this program overview section. Like you will be getting an opportunity to practice the like hands-on experience with the data sets available on the public domain, to know about the like phylogenetic interactions between hosts and the viruses. So coming down more, you can see that there is an interactive calendar, we can say, with some of the dates which are highlighted. Like today, 17 is highlighted. On that particular day, like on the dates which are highlighted, on these particular dates, we will be having a live sessions. So let's say like today we had a first session and we have learned about the next generation sequencing, viral genomes in host transcriptome, and what all different topics we have covered. You can see here like types of infectious diseases, then recent publications, then types of NGS data you all can go through. Apart from that, you can see that there are some of the associated online resources which you can go through to know more about the topics. And similarly, our next session will be there on Feb 21st, and we will be covering the multiple sequence alignment and phylogeny part. So coming down more, as you scroll down, you will see that we have three different tenures. One is for 45 days, 60 days, and 90 days. So the like common resources or the common outcomes for all these three resources is you will be getting the online tutorials, you will be getting a chance to work on the project examples available on the Learn portal. Also, you will be getting a hands-on coding sessions, and by chance, let's say if you miss some of the live sessions, so no need to worry about that, we will share the session recordings as well. Apart from that, you will be getting online mentor and technical support, and once your program is over, then we will provide you with the certificate of completion, and along with that you will be getting the access of TE Info server. So for the 60 days, what extra you will be getting is you will be getting a one-on-one mentorship session. And while going through the courses, let's say you come up with your own research idea to work on. So you can like even propose your research question, and you can like share it with the mentor. And along with that, you will be getting an opportunity to like present your research proposal as a in the form of a poster in various international symposiums level. And in the 90 days you will be get the like license, and one-on-one mentorship will be there, and you will get an ample amount of time to work on a research question, and like you can like properly perform your research work, and you will be getting a chance to get it published in the peer reviewed journal. So these all are the resources and the outcomes for all these three different tenures. Coming to the pricing part, as you all can see on the screen, that for 45 days we have INR 7500 for Indian and Asian customers, we can say, and for 60 days we have like 9999, and for 90 days it is 14999. Let's say like some of our participants are from African region, so here you all can see that there is a drop down So just click on that and you can select this thing like Naira NGN. So you can see that the pricing have changed to Naira. Like for 45 days it is 49,999, for 60 days it is 99,999, and for 90 days it is 149,999. And let's say some of the participants are from US or some other countries, so they can also see their pricing similar to this in USD also, like $150, then $300, and $450. So how to enroll for this particular program? So let's say I'm interested in the 45 in 90 days tenure. So what you have to do is just simply click on the buy now button, and you will be directed to a checkout page. Like this is your checkout page. Yeah. So you will be like reviewing your order, like the program. You just simply have to fill up your user details, name, email ID, then your education, and after that don't forget to click on this checkbox. And after that you can select any of the payment method like Stripe or PayPal, or if you're facing any issue you can like mail us. We have alternatives also like bank transfer, and for Indian customers we have like UPI option is also there, and we have alternative methods for the payment part. So this was all about the registration and enrollment part. If you have any questions, please feel free and I'll address those things. Thank you.
Thank you so much, Shubham. So, does the participants have any queries regarding the registration or uh, regarding the enrollment? Please let us know and we'll be happy to answer. We'll pause for a couple of seconds for the participants to share their queries. If you wish to receive, for those participants who have not enrolled yet, but want to get more details about the program as well as the scholarship details, kindly share your email ID in the chat box so that we can mail you regarding details. Thank you so much, Uluvaimi. What about the rest of you? If you have any queries, please share your uh, queries in the chat and we'll be happy to answer. Thank you so much. I can see that the participants are sharing their email ID. We will be taking a note of all of that. Yes, Uluvaimi, we'll send you the scholarship details on your email ID that we have shared. Shubham, we have a query from Sunil. He is asking, are there any fully funded scholarships for the program?
Unfortunately, we do not have any such like fully funded scholarship, but yes, like up to like 50% scholarship is there. So like you can mail us or you can share your email ID. We will share you with the like details, scholarship details, and the scholarship form, so that you can fill up that form and we can provide you the scholarship. Or else you can contact us at uh, Yeah, so I have shared the contact. You can mail us at marketing@omicslogic.com, or you can message us on WhatsApp. The number I have provided is active on WhatsApp. So we will provide you with the scholarship details. And those all who have shared their email ID, we will also be providing you with the session recordings and the scholarship part. Thank you.
Thank you so much, Shubham. Do we have any further queries? If so, please share. And if anyone is left to share their email ID, please do so. Otherwise, we'll be ending today's session. All right, then if everyone has shared their email IDs, we'll be ending today's session. So thank you so much, everyone, for joining. And the program participants, if you have any queries, please feel free to reach out to us through the WhatsApp group or our email IDs. And for those of you who have not enrolled, we look forward to seeing your registration and in the upcoming sessions. Thank you so much, everyone, for joining, and have a great day.
Thank you. Bye.